Hello everyone and welcome to yet again another very entertaining video. Today's topic is the regulation of the metabolic hormones T3 and T4. And I start with this attractive picture of a goiter. Goiter is one of those words that just rolls off your tongue. It's fun to say. And the etymology is gullet, or even going back even further, Latin, throat. And goiters have been around for a long time. I think the earliest records of goiters from a medical point of view would be back in Chinese literature dating back to about 2600 BC. And back in the day, they used to treat it with seaweed, which actually worked because seaweed is fairly high in iodine. Of course, what the goiter is an enlarged thyroid gland. Let's draw a proper thyroid. The thyroid gland is this butterfly-shaped gland. And we've already talked about some aspects of the thyroid. The parathyroid glands, positioned on the ventral surface. You can't really see them from the front, but we'll kind of draw their general location. Involved in calcium, and the thyroid itself involved in calcium regulation, opposing what the parathyroid glands are doing, parathyroid hormone. But there's another part of the thyroid that we want to talk about today. If we were to zoom into the thyroid gland, we would see a couple of different structures. Let's get rid of the parathyroid. We would see larger structures called follicles, and in between the follicles we would find what are called parafollicular cells. These are the cells that we're familiar with, as they're responsible for the release of calcitonin, which antagonizes the effects of parathyroid hormone. But today's video focuses on the follicle and what these are doing in the regulation of metabolism. Now that we've kind of set the stage for where we're at, we actually need to back up and look at this from an endocrinology point of view. And like other major endocrinology signaling pathways, this one too starts up here in the brain with the hypothalamus. Now remember, the hypothalamus is going to signal through the hypophyseal portal system to the anterior pituitary. And for the hormones that we're discussing today, the releasing hormone from the hypothalamus, the chemical signal being released from the hypothalamus, is called TRH. This stands for thyrotropin releasing hormone. TRH will enter the hypophyseal portal system, exit the other end on the anterior pituitary, and cause the release of TSH, which stands for thyroid stimulating hormone. TSH is released into the blood, and its target is the thyroid, specifically the follicular cells. And the thyroid, once stimulated, will release two hormones, T3 and T4. We'll just kind of group them together. And as we've talked about previously, it's important to understand feedback. T3 and T4 have the role of playing negative feedback on both TRH and TSH. So there's the signaling axis. And importantly, what do T3 and T4 regulate? These regulate the metabolic rate of the body. In other words, T3 and T4 are all about metabolism. All right, now that we have a general sense of the signaling axis and its negative feedback, we want to take a journey into the follicle itself and see how this process happens. Now, if we were to zoom in on one of these follicles, we would see that it's actually a round structure and hollow inside, and that the follicle cells that we're interested in make up the outer shell of this hollow structure. Now, hopefully you're asking, why is this thing hollow? And we're going to show you a very good reason for that. Before we do that, let's take one of these cells. Let's take this cell right here, and let's expand it and see if we can't understand the process of creating T3 and T4 and then understand the regulation by TSH. All right, we've isolated this follicular cell. Let's expand it. And just to orient ourselves, remember this is the outside of the follicle. This is the inside space on this surface, what we will call the luminal surface. And one of the things we want to know is, why is it important that we have this hollow space in the middle of this follicle? And we'll address that in just a second. Now, to start with, T3 and T4 are actually amino acid-derived hormones. In fact, they're derivatives of the amino acid tyrosine. Now, going back to our basic understanding of cell biology, proteins are made in the rough ER. And that's where we're going to start today's journey. So we've got our nucleus, and of course, its outer membrane, its second membrane is continuous with the endoplasmic reticulum. And here we have the rough ER studded with ribosomes. And this is where proteins associated with the endomembrane system, proteins that are going to be associated with the membrane or proteins that are going to be exocytosed, this is where those proteins are going to be created. And the protein in particular that we're interested in, it's not really going to be folded and made into any specific protein. It's more just a polypeptide chain. Is something called thyroglobulin. Now, as we go through this process, let's make a list of things that we want to remember. One of the things that we want to remember is this. Thyroglobulin is enriched for the amino acid tyrosine. Now, to understand why that's important, let's draw the amino acid. Of course, the basic amino acid structure is going to be a nitrogen, a central carbon, a carboxyl group, and then the side chain for tyrosine looks like this, where it's got this aromatic 6-carbon ring on it, and then a hydroxyl group down here. Keep that image in mind. We're going to come back to it. All right, so thyroglobulin is made inside the rough ER, and then it buds off. Of course, it's going to go through the Golgi and all that stuff, but it buds off and it's ready to be exocytosed. And we're going to exocytose the thyroglobulin into the lumen. In other words, we're going to go to the luminal surface for this. So this will come to the membrane, and with exocytosis, the membranes fuse, and these polypeptide thyroglobulin chains, consisting around 80 amino acids and enriched in tyrosine, are released into the lumen of the follicle. Well, now we can answer why the lumen is so important. In order to make T3 and T4, we need to undergo a fairly nasty reaction. We need to attach iodines to these molecules. Now, in order to be reactive, we need to use iodine, but iodine is not really stable. Of course, as we take it in our bodies, it's iodide, the stable anion of iodine. Now, conveniently, this cell is really the only cell in the body where we're going to use iodide. So, how do we get it into the cell? Secondary active transport. On the outer surface of the cell, we have a transporter called NIS, sodium iodide symporter. And here we're using the strength of the sodium gradient to also bring in iodide, I negative. Again, iodide, perfectly safe, non-reactive. So let's take that iodide and let's let it leak out into the lumen. Out here, it's going to encounter an enzyme called thyroid peroxidase. And in a redox reaction, thyroid peroxidase is going to take my cation, I negative, and turn it into the very reactive iodine. And iodine is going to attack the aromatic carbon rings on tyrosine. And again, just remember, this is all in a controlled environment. That's why the lumen of the follicle is so important. I don't want to be doing this anywhere, but in a controlled, isolated environment, I can deal with the nastiness of this reaction. Now, what does this look like in tyrosine? We've got one of two possible outcomes for tyrosine. The ring could be iodinated once, and this would lead to a structure we call colloquially a MIT, which stands for monoiodotyrosine, or it could be iodinated twice. And if we put that second iodine on, now we have a DIT, or diiodotyrosine. So these thyroglobulins are being loaded with MITs and DITs. The tyrosines within the thyroglobulin are being bound to this reactive iodine to make MITs and DITs. And if maybe we were to zoom in on this molecule, I hope you can appreciate, we've got this long polypeptide chain, and it's covered in these MITs and DITs. Well, the next thing that's going to happen is these MITs and DITs are going to start interacting with each other. And let's take a closer look at what that looks like. So here we've got a picture of the MIT and the DIT. And as they start to interact with each other, they start to exchange or share these aromatic rings. And here again we have two options. If we add a MIT to the bottom of a DIT, we create T3, which stands for triiodothyronine. The key feature here, recognizing T3 has three iodides, 1, 2, and 3. And if we add a DIT to a DIT, we get T4, which is called thyroxine. So we've just made our T3 and our T4, but as we go back and look at the follicle, remember, these T3s and T4s now still are bound to this thyroglobulin polypeptide chain in what we call the colloid, which is the substance within the lumen of this follicle. Now we need a trigger to release it. That trigger comes in the form of TSH. TSH is a ligand for G-protein coupled receptors. This is the TSH receptor, and we're not going to detail G-protein coupled receptor signaling, but if you recall, this is going to activate an alpha subunit, and in this system, alpha subunit targets adenylate cyclase. Of course, from previous units, we know that adenylate cyclase takes ATP and converts it to cyclic AMP. And through a signal transduction mechanism, this is going to lead to endocytosis, where we're going to bring into the cell these thyroglobulin molecules loaded with T3 and T4. We're then going to fuse these endocytic vesicles with a lysosome. Remember, lysosomes are acidic, they contain hydrolytic enzymes, and with this fusion, we're going to break up the polypeptide chain that was thyroglobulin into individual amino acids. Of course, we want to recycle all the other amino acids. So amino acids back up to be used, but my T3 and T4 are now liberated from this chain and ready to be used. And to get them out, we have an ATP-driven primary active transporter called MCT. It stands for monocarboxylate transporter. And this is going to actively pump T3 and T4 out into the blood. Now, because of the large aromatic rings on these molecules, they're fairly hydrophobic. So in the blood, we've got carriers that will bind them and help them as they travel through the body. Here's T3, here's T4, surrounded by carriers. And remember, one of the key things about carriers is we're going to increase half-life. And this is especially relevant for T4, which can be in the body for several weeks for use by nearly every cell in the body as a means of regulating metabolism. Now, the last thing we want to talk about is what happens next. So we're in the blood, and we've got T3, we've got T4. T3 is shorter lived. It's got a fairly short half-life, hours, whereas T4 has a long half-life. However, T3 is biologically active, whereas T4 is mostly benign. So T4 represents my reservoir, whereas T3 represents the bioactive molecule. Now, for any given cell, we're going to typically transport these into the cell through that same MCT transporter. As T3 comes in, it's going to use intracellular receptors, and we talked about these in a previous video. We're not binding to receptors on the outside, we're coming straight in. And the analogy we make for how these works is a car without a driver. As T3 comes in, T3 acts as the driver, and what's the road? DNA. This activated car now acts as a transcription factor to transcribe all of the genes associated with metabolism in that cell. Of course, T4 could also come in, but remember, it's not nearly as bioactive, but as a reservoir, it's super useful because if the cell needs it, individual cells could have an enzyme called deiodinase where they could take that T4 and simply convert it to T3. And now we're off and running. So there's the final chapter in the story of the production and use of T3 and T4. These are very important metabolic regulators, as seen by their deficiency or overexpression. High levels of T3 or T4 could lead to hyperthyroidism, which is going to be associated with increased metabolism and all of its various symptoms. Weight loss, fatigue, intolerance to hot weather, whereas hypothyroidism would simply be the opposite. Weight gain, low metabolic rate, still fatigue, and intolerance to cold weather because of their inability to generate heat by themselves. And there are various causes for why this might occur. So in summary, let's go back up to the signal axis. The release of T3 and T4 are the regulatory mechanisms. If T3 and T4 are low, then the negative feedback on TRH and TSH is lessened, and TRH then stimulates TSH to stimulate the thyroid to make more T3 and T4. Once I have enough T3 and T4, the negative feedback inhibits the release of TRH and TSH. This is how I maintain a balance with these important metabolic regulators.